Hi everyone, welcome to Gina Editor's YouTube channel. I'm Pooja from Krista Jayanti College and I'm going to be briefing about lactoperon. So, introduction. E. coli can use lactose as the carbon source. The enzymes required for the use of lactose as carbon are synthesized only when the lactose is available as carbon source. So operon is an operating unit which can be defined as cluster of genes located together on chromosomes and transcribed together. It is a group of closely linked structural genes and associated to control gene which regulate the metabolic activity. All the genes of an operon are coordinately controlled by an enzyme lactose mechanism, lactose operon, first described by Francis Jacob and Jacques Monod. So, lactose operon is an operon required for the transport and metabolism of lactose in E. coli and many bacteria. So, operon or a group of gene with a single promoter that encodes gene for the transport and metabolism of lactose. So, the operon consists of cluster of genes that encode proteins to facilitate a common metabolic pathway. So structure of lacoperon. So the lacoperon usually consists of three structural genes that is which are used for transport and metabolism. So these structural genes are LAC-ZIT, LAC-Y, LAC-A, which will encode beta-galactosidase, lactose-permease, and thiogalactosidase transacetylase. So all three structural genes are under control of promoter P along with all other control element operator. Operator is O. Binding of activator enhances binding of RNA polymerase to promoter, preventing transcription. Binding of repressor protein to the operator blocks binding of RNA polymerase to promoter, preventing transcription. Lac-operon is controlled not only by LAC promoter or LAC-O, also by separate regulatory gene LAC-I, that is catabolite activator protein, that is CAP, acts as glucose sensor. The CAP binding site is the positive regulatory sites. Now, next is LAC repressor. The LAC repressor is a protein that represses transcription of LAC operon. It does this by binding to operator, which partially overlaps with promoter when lactose is not available. The LAC repressor binds tightly to the operator, prevents transcription. When lactose is present, the LAC repressor loses its ability to bind DNA. It floats the operator creating. This is the structure of lacoperon. Gene expression of lacoperon. So we have two expre- two negative control and positive controls. Now we're gonna see about negative control of lacoperon. A negative system to turn off the lacoperon when lactose is absent or when glucose is present. In the absence of lactose, the regulator gene produces repressor protein, which then bind to the operator site and it will prevent transcription. If it prevents transcription, as a result, what happens is it will not produce any mRNA and thus proteins will not be found. That is what in the image it is shown. When lactose is absent, the LAC repressor binds tightly to the operator. It gets in RNA polymerase way, preventing transcription. Now we'll see about the positive control of lacoperon. In positive control, what happens is it will turn on the lacoperon when lactose is present and glucose is absent. So when lactose is introduced into the medium, it will bind to the repressor, but the repressor fails to bind to the operator. What happens is, therefore, the operator is made free and it will induce the RNA polymerase to bind to the initiation site of the promoter, in which it will result in the synthesis of mRNA. So this mRNA, which will code for three enzymes necessary for lactose catabolism. That is what is shown in the image. So when glucose is absent, lactose is present. So next is the CAP and CAMP. CAP is catabolite activator protein and CAMP is cyclic AMP. So E. coli contains a protein called CAP encoded by a protein called CRP, and a cyclic AMP is synthesized enzymatically by the enzyme adenyl silase. So when bacterium grown in culture medium containing glucose, the cyclic AMP concentration of the cell is quite low. When the same bacterium is grown in the culture medium containing glycerol or other carbon source, the concentration of the cyclic AMP will become high. So when lactose is present, the lac repressor loses its DNA binding ability. RNA polymerase alone does not bind very well to the lacoperon promoter. Cap binds to the region of DNA just before the lacoperon promoter and helps RNA polymerase attach to the promoter, driving high levels of transcription. Cap isn't always active. Instead, what it will do is it will regulate by a small molecule called cyclic AMP. So cyclic AMP is the hunger signal made by E. coli when glucose levels are low. So CAP is only active when glucose levels are low. So there are four scenarios where glucose is present or absent, or lactose is present or lactose is absent. So now first one is glucose present, lactose absent. So when glucose is present, lactose is absent, no transcription of lac operon occurs. That's because lac repressor remains bound to the operator and prevents transcription by RNA polymerase. Also, cyclic AMP levels are low because obviously glucose levels are high. So cap is inactive and cannot bind to the DNA. See, as you can see in the image, that is what it's explained. So next is glucose present, lactose present. So when glucose is present and lactose is present, both are present. So low level transcription occurs. The lac repressor is released from the operator because the inducer is present. So cyclic AMP levels, however, are low because glucose is present. Thus, cap remains inactive and cannot bind to DNA. So transcription only occurs at low leaky level. You can see in the structure. Next one is Next one is glucose absent, lactose absent, both are absent. No transcription occurs. Cyclic AMP levels are high because glucose levels are low. So CAP is active and will be bound to DNA. However, the lac repressor will also be bound to the operator, acting as a radio block to RNA polymerase and preventing transcription. This is the image. So next one is glucose absent, lactose present. Strong transcription of the lac operon occurs, the lac repressor is released from the operator because the inducer is present. So cyclic AMP levels are high because glucose is absent. So CAP is active and bound to the DNA. CAP helps RNA polymerase bind to the promoter, permitting high levels of transcription. So this is the image. So these are my references. You can refer to these links for more information. Thank you.